Well, thank you for the invitation to come and talk about this today. This is I've been teaching this specific topic for over 35 years, mostly to college juniors and seniors, some freshmen and sophomores, but in general, it's the standard sort of botany 101 kind of a thing. But we're going to look specifically today at understanding and educating people on how the fine details of flowering plant reproduction work functionally. Most people can see a flower and know the general parts and basically how the flower operates. But what I want to do is look at a smaller scale and look inside the flower structures to explain what's going on and compare that reproductive process to one you may be a bit more familiar with. So with the interest of looking at people and their interests with cacti and succulents, I've chosen examples that may be a bit more familiar to you with respect to the plants that we're looking at. So I want to start by talking about what we're not going to cover. And this is asexual reproduction. So some people will know this as clonal propagation, fragmenting the plants into smaller pieces that naturally dissociate. Offsets that will form through normal vegetative growth and reproduction, sort of like the pups people call them, or the babies that form around the base of Haworthia. Those are not really babies. They are just clonal offsets or clonal propagations that the plant does by means of its normal growth and development. Asexual reproduction is a safe way of propagating, simply that there's no risk or no chance involved. It's basically taking the plant material and making an exact genetic copy of that source plant. And from the evolutionary perspective, it really does nothing to enhance or improve a natural population, other than making the plant, that genotype or that specific genetic variant of that species persist for a longer time, and that in itself may enable more and more opportunities for the plant to reproduce in the future. So it's not completely worthless in the evolutionary sense, but there's no innovations or no novelty in asexual reproduction. And because asexual reproduction is a local event, it happens locally in nature, with the exception of some dispersal type things that you would say an Opuntia pad getting stuck to the backside or the fur of an animal moving through a region that can travel very far. Many asexual propagations are not really very useful for moving the genotypes or the evolutionary units through the landscape. Okay, we do have exceptions. Many of you know about Opuntia. That's as one of the sort of the poster child for clonal propagation, because each one of the stem segments, the cladodes, can break apart and root, and we take advantage of that many times in propagating the plants that we love, which are the succulents. So to many of you, asexual reproduction is just a standard mode of duplicating or making more copies of the same individual plant, that sort of clone, as people will call it. And this is something that has been going on for decades, for a couple hundred years easily. Going back to the 1700s at botanical gardens, when they would make clonal propagations of plants that were particularly difficult to obtain. So what I want to do today is look more specifically at sexual reproduction of plants, and in this case, it's going to be an overview of how flowering plants will reproduce on a normal basis. There are always some variants. Nature has added a number of different modalities to the general process, but we're going to look primarily at the floral parts. We're going to look at the structures and so on. And I want to compare those to a reproductive cycle that some of you, in fact, hopefully all of you, are aware of, because this is your reproductive cycle. You exist as adults, and then certain tissues in your body are capable of taking your what we call the diploid genome, the two sets of chromosomes, and reducing them through a process we know as meiosis directly from this adult organism to the gametes, or the compatible haploid 1N. Each of these units has a single set of chromosomes, reducing those from 2N to 1N through that process of meiosis. And that allows the gametes to be formed directly. We call it a gametic meiosis. And then through various ways, animals allow the sperm and eggs to come together in different manners. There's behaviors involved with this in many animals. In some, it is a random event where, say, if you have corals or mollusks growing and living in the ocean, the gametes are simply dispersed into the environment, into the water, and it's just a matter of luck whether the sperm finds an egg cell to fertilize it. So one way or another, the sperm will find the egg, and then eventually the chromosomes in the sperm fuse with the egg cell chromosomes, and we wind up with this 2N cell, the first cell of the next generation. And then through the process of embryogeny, forming a juvenile, and of course then through maturation, the cell division process of mitosis simply increases the size of the individual until it reaches maturity. So this is the standard growth and development cycle that we are familiar with, and it is one that virtually all animals share. There are variants, but animals have this type of growth cycle throughout its annual or lifetime number of growth cycles. Plants are a little different. Plants do not do that relatively simple procedure. They have what we call an alternation of generations life cycle, meaning that there's two different phases of the life cycle, each of which is somewhat independent. So what I'm going to do is walk you through the difference in of the life cycle you just saw, and then we're going to understand that life cycle, I hope, and then we'll look at all of the parts that make that life cycle happen. Okay, so here we have a typical flowering plant life cycle. This is going back, this is basically Botany 101. And here you can see, we assume that the flower occurs on a mature plant, a mature adult plant that has a sufficient size to withstand all of the metabolic energy that it takes to make flowers. To do this entire process, plants have to have a certain critical size at which they can actually sustain flowering. So this is why juvenile plants generally will not flower until they accumulate enough biomass, enough metabolic energy, shall we say, in that plant to be able to devote the energy to making flowers or inflorescences and then producing the structures to complete the sexual reproduction life cycle. So we'll assume that the flower is achieved on an adult plant. And of course, on that plant, that there are sterile parts. There's on many flowers, there's a sepal, a whorl of sepals down here. We call it the calyx. There are petals on many flowers, of course, not all, but there are petals on many flowers. Some of these can be very showy and attractive, which is another reason why we grow the plants that we do, because of the beauty of some of the flowers. But those are sterile parts. There's no gametes involved with that. There's no formation. There's no of that meiotic process of going from two sets of chromosomes to one. The only sort of business end, the action end of the flower, occurs either in the anthers, which are the male parts of the plant, and the anther is specifically just this little structure just at the end here. So this is the anther, and the anthers are supported on this sterile stalk of tissue we call a filament. So the only place in the male part of the plant where meiosis can occur is in that anther. We'll look then at the female over here. If we look at the female, we have the whole structure, which we call the pistil. You may be familiar with that term. That pistil consists of three parts. At the top, there is the receptive part, which will receive the pollen. We call that the stigma. Here you have the style, S-T-Y-L-E, and then down below, the place inside that contains an empty space, an open space. We call it the locule. The ovary is the wall, and the space inside is where the specific female structures occur, the ovules, which are the presumptive seeds. If the reproductive process takes place fully, that ovule eventually will turn into a seed. So we have diploid parts here right now, and we haven't talked about the process that we know as meiosis, reducing that chromosome number. So here you can see up in the anther, you have cells that are up in these 4 parts, we call them locules. Those diploid cells undergo meiosis from 2N diploid down to 1N. So each of these individual smaller cells has one set of chromosomes. That is eventually going to develop into a pollen grain. So each of those 4 products of meiosis will separate and then get a wall built around it, which allows the pollen grain to leave the anther into the environment and still not dry out. It has a material around it that's deposited by the plant called sporopollenin, and it is a rugged material. It reduces the chances that the pollen will dry out as it makes its way from the anther to somewhere in the environment where there may be a receptive stigma. It could be 1 meter away from an adjacent flower, or it could be 1 mile away on the legs of an energetic bee. So the pollen grain has to be intact and be able to withstand whatever trip is being made. On the female side, inside that ovary, that ovule that sticks into the center, I'll show you some photographs of that later. Those ovules contain a cell that undergoes a cell division from 2N to 1N. And like over here, the process is that 4 products will form, but only one, the so-called durable or the persistent megaspore, will continue. And through some additional cell divisions, make a set of nuclei in here, inside this structure, which is, of course, inside the ovary. And flowering plants are really special in that they have 2 phases of pollination. 1 phase of pollination, 2 parts of fertilization. And I'll talk about that in a second. Inside this female part, there are nuclei that are receptive to other sets of chromosomes that would come from the male part. But although this looks very complicated, it's really quite simple. Only 2 groups of these smaller cells, these with haploid nuclei, 1N nuclei, are going to be able to accept the chromosome set from the male. So once you have this as at a sort of a ready and waiting state, and if you have a pollen grain that can be released, the next phase of this whole process is to somehow move that pollen grain to the stigma up on top of the plant here. So that is what takes place with a bee, a bird, a bat, or a brush, okay, if you're pollinating these by hand, manually. Once the pollen grain lands on the stigma, it will, like a seed almost, it will germinate. The cell material inside that pollen grain will start growing out almost like a little root. But instead of being a root, it grows down, not into the soil, but that pollen tube grows down into the tissues of the female plant, the style here, and then grows down and enters the ovary and delivers inside of that little tube, that pollen tube, delivers a set of chromosomes. We call it a sperm nucleus. And it delivers that to one of the cells down here, the so-called zygote that forms from the fusion of the egg cell and one of the sperm nuclei. The other sperm nucleus that is here fuses with these central cells, and you get a fusion in the middle here, a 3N, what we call the endosperm. Now, this process is the standard method, the way that actual fertilization takes place. So pollination is fertilization, is the actual fusion of the nuclei with the cells that were over here in that so-called mature ovule. Okay, so it's double fertilization, 2 nuclei fuse. Once that happens, then you have this zygote. That's going to be the next seedling. That's the 2N juvenile form that will continue to form the embryo. And seeds are nothing more than the embryo packaged inside with some storage material, whether it's packaged adjacent to the embryo, like you would have in a grass or some other monocotyl, the so-called endosperm tissue, or if the embryo is going to be supplied with nutrition, the plant supplies nutrients directly into the embryo in the form of these things we call cotyledons. So the seed is simply packaged with the embryo, the next generation, a bit of part of the generation. So this varies extensively from that simple, go back here, from this relatively simple life cycle. There are actually two phases of the life cycle. We call one is the sporophyte phase, and the other is the gametophyte stage. So we have male gametophytes, we call microgametophytes. Let's say all the parts are small, and the megagametophyte, all the parts are large. So this life cycle that we have in all of our flowering plants alternates back and forth between sporophytes and gametophytes, sporophyte and gametophyte, and they follow this cycle throughout their evolution and development. So this is I've tried to make it a little bit less complex than it looks, but there is more to this life cycle than the one that you have successfully navigated so far. So what's the big deal about sexual reproduction? Why is it important that we don't keep clonally reproducing plants, breaking apart and starting new plants, right? That would be really easy. Well, evolutionarily, sexual reproduction is really important because it helps increase and maintain the genetic variability in the populations of organisms, whether it's a cactus, whether it is a bat population in Malaysia, whether it's a set of algae that's growing on the side of a rock in a river, whatever the population of organisms is, if those organisms can reproduce sexually, there is a good chance that genetic diversity, having a wide range of the genetic potential to survive in a wide set of ecological challenges in the environment, if that plant can maintain or in fact increase the genetic diversity in the population, it stands a better chance of survival because it has the potential to adapt to that environment. This is Charles Darwin's basic premise on natural selection. So sexual reproduction and the ability to exchange genes within the population is a huge thing evolutionarily. So because of that, no one, I think, is going to disagree with this, whether you are a person, a politician, or a botanist. Sex is good. It doesn't matter. Sex is good because of the potential for maintaining this genetic diversity. That's why it's important to have this process as part of the life cycle for any organism that's out there. Even bacteria, which don't have true sexual cycles, have the ability to do a sort of pseudo-sexual cycle by exchanging small particles or small bits of DNA, and that also enhances their genetic diversity. The problem that some growers have, whether you're growing begonias or roses or cacti or aloes, whatever it is you need to know the names and understanding on how those important parts work. So here's one of my students sent me this some years ago, and he says, be careful, don't get pollen on your stamens or you'll get pregnant. Well, the artist that composed this never had a lesson in plant biology or a webinar like this, because you know that pollen actually comes from stamens, and they probably meant that they don't want to get pollen on their stigmas. So you need to know the parts, and this is the big deal. The other thing is that with respect to plant reproduction, plant sexual reproduction, there's no emotions involved here. There's no, it's pretty much a perfunctory, sterile thing that takes place in the absence of any behaviors like you would have to have in animals, and have the expression, the interchange of different kinds or different units within the population. You have certainly the same kind of effective receptivity. There are only times when pollen will be accepted by the stigma, when the stigmas are ready, because those ovules inside the ovary are mature. And there's a certain timing of when the pollen is ready. And you're probably aware of that from trying to do propagations in your own plants. And of course, the parts of the plants are vastly different than you have between plants, plant structures that we just reviewed in the flower versus other parts that might be present for reproductive purposes. So going back to those parts, that just jumped on me there. Again, much of this is going to be not new to most of you, but the business end, as I said before, are not the sterile parts. It certainly focuses specifically on the female parts here in the ovary, the style, and the stigma, and then the anthers, which are generally presented to whatever the pollinators are by those filaments. Okay, so those are the fertile parts, the stamens and the carpels. The stamen, by the way, is simply the combined structure of having both the filament and the anther. So the anther, very specifically, is just this end part right out here where that meiosis can take place. So this is not an anther down here. This is the filament, but if you want to call them all stamen. Okay, so that's just clarifying some of the terms that you may be familiar with. This kind of reviews that in what we call floral whorls. A whorl is nothing more than an arrangement of plant parts that arise from a similar location along a plant axis. So you have the sterile parts, the calyx and the corolla, the sepals and the petals, respectively. And if you take all of the anthers to male house and the pistil, which is collectively all of the parts that are female, called the gynoecium, some slightly different to the North American pronunciation. Flowers themselves can have parts. So in this case, on the left, you see what we call a radially symmetric flower. And what that literally means is that, so you have bilateral flowers that have mirror image symmetry. We call them zygomorphic flowers. And this has to do with how the pollinators perceive the flowers. We see both actinomorphic and zygomorphic flowers that are present in the plants that we love. Mesems and cacti certainly have them, but cacti also have zygomorphic flowers. These are adapted more for insect pollination on the left, and the flowers that are adapted to hummingbird pollination generally tend to have the syndrome of red flowers with a tubular corolla, or the fusion of, or presentation of the petals. Same thing down here with Haworthia. The way that the insects perceive the flower is basically how the floral parts are presented to the pollinator. All of the parts are generally found on most flowers. If both anthers and pistils, the gynoecium, are all present in the flower, we call it a perfect flower. Some flowers are unisexual, like we have in euphorbias, like you have in Didieriaceae, for example, where there's only going to be male flowers or female flowers. In some cases, both male flowers and female flowers can be found on the same individual plant, but in different places. And in others, there are male plants and female plants that are separated. So it really depends on the group you're looking at with respect to how the floral parts are presented. There's also positions within the flower where the ovary is above the parts that are attached. We call that a superior ovary, or if the ovary down here is below where the other flower parts attach, we call that an inferior ovary. Sometimes you'll see these in some of the books and articles, publications that are presented about our plants, and this is where these terms come from. Internally, the parts of the plant, the male parts in the flower, if you look here, if you take a slice of that part of the flower, this is what you would see at the cellular level. So in this case, we have 1, 2, 3, 4, 5, 6 anthers around the top of this. And down here, if you slice through the ovary, you see the ovary wall. You see these little projections into the space. These are the ovules where the seeds that will eventually become the seeds. And inside of there, you can see right there, that is an egg cell that would be present inside of that ovule. So it's important that in the flowers that the path for the pollen to arrive, the path for the pollen nuclei, the so-called sperm nuclei to be delivered, needs to come in and then enter into that ovule through a specific place, that opening that's right there, we call the micropyle. So in the male parts, we've already talked a little bit about this, that the stamen is composed of filament and anther, and that anther provides for the release, we say, the dehiscence of pollen when the pollen is mature. You can't coax immature pollen out of an anther. It's essentially worthless to do that. So when the pollen is ready, usually the anthers become very plump, and then you would look for, in most plant groups, some sort of a slit or an opening down the side of that anther that you would see right there. That is an indication that the pollen is ready to be released. On the female side, if you continue and look at the ovary, the stigma is the sort of the business end of that female structure, that gynecium, which is the place that receives the pollen. And there can be all different kinds. I'll show you in a second. There's all different kinds of receptive areas, but in general, those areas often will exude a sticky liquid material. Usually it's some form of a sugar that is released, although there's, in some plants, it's actually a gum that's present, a different class of chemical compound. But in any case, the stickiness of that will allow the adhering of the pollen to that, and also allows for the rehydration of the pollen from liquids from the female part into the pollen grain, so that the growth of that pollen tube can be activated. That's the way it happens. Okay, so there's a lot of variation that's in flowering plants, and in most cases it is associated, the stigma and the structures associated with it are relevant to the kind of pollinator that does the pollinating. And then ultimately the function of those pistils is to grow and develop into the fruit, whether the fruit is dry or remains fleshy at maturity, and that also is going to vary by plant group that we're talking about, often varying at or within the family level. So the female parts can have all different styles or different kinds, I should say styles as a pun, but there are different forms of the ovary down at the base, as well as the stigma and style that delivers the pollen tube into the ovary chamber. But you'll notice there's always some form of an opening or chamber inside of which those ovules can form. So these ovules now are growing sort of out into the space. You can see them in here in a smaller ovary with the smaller spaces, but still those ovules are present everywhere. Those are presumptive seeds, but just because an ovule forms doesn't mean it becomes a seed. It has to be fertilized, okay? And to do that, the pollen tube has got to grow down toward the ovule and then grow into the tube, like that little root would be growing down through the soil. The pollen tube grows down through the female tissues into that space and then grows into the center part of this ovule. So this is the outer walls. This is the, we call this the funiculus. It's held against the wall of the ovary, but this grows into the space that is created inside of that ovary that you just saw. And there are basically two places where those sperm nuclei fuse. One is to the egg cell right here, and the other is to these, what we call the polar nuclei, so that one nucleus comes in and fuses with the egg cell, the other nucleus comes in and fuses with this central cell and fuses all three polar nuclei together. 1N plus 1N plus 1N from the sperm equals 3N, and that forms the so-called endosperm tissue. And this happens in virtually all angiosperms. So there's individual ovules at maturity. The flower is dissected at the ovary, and you can actually see the little tiny micropyle right there. I'm circling it right now with the pointer. That is the place where the pollen tube would grow into. Here are some pictures of ovules of lithops that I took while I was doing my PhD work out at Rutgers University. And this is the ovary wall out here. We call it the placenta because that's where all of these stalks, these funiculi attach. And here is an ovule. And you can see the opening right here. That's the so-called micropyle. The pollen tube will have to grow up and into that structure right there. And then underneath this outer skin, the outer integument, we call it, there is actually some patterning of the cells that becomes the outer part of the seed coat. That's what you will see when these ovules are mature. So this is from the female side where this reproduction takes place. So what we have seen so far is the development of pollen in the anther. And we'll talk next about the delivery of the pollen. We have talked about the ovary and the ovules that grow into that space inside the ovary, forming that receptive ovule structure right here. We call that whole structure inside the megagametophyte right there. Some people will call it an embryo sac, but there's no embryo yet. And we've already just talked about the pollen tube growing down through the tissues of the stigma style and into the ovary to fuse with those cells that are on the inside here. If that happens and fusion takes place, you would have the sperm forming a zygote, a 2N cell that eventually turns into the embryo inside that ovule. And as that ovule matures, it deposits the seed coat around the outside, which are then released into the environment and helped in its dispersal by the fruit. So it's the fruit, the function of the fruit is to disperse the seeds, period. It's not for us necessarily to enjoy that. We grow many, many fruits for our use as food, but evolutionarily the function of the fruit is to help disperse the seed, one way or the other. Once those seeds are put into the environment, they can take up water. They can germinate, which means germination simply means by taking up the water, it activates the growth and development of that embryo, which is kind of shut down until the seed receives all of its necessary environmental cues, which means nutrients, water, air, and proper temperature. If you can supply all of those, you'll get germination. And then, of course, you know that the germinating seed grows up to an adult plant to start the whole process again. So floral presentation, the actual flower is presented to attract pollinators because this is one of the uncertain steps in this sexual reproduction process. You can present flowers individually or in clusters. We call them inflorescences. And the whole role of that is specifically to attract pollinators to co-opt them into moving the male gametes, the pollen grains, to the female receptive structures. So the plant really has evolved over the last 110 million years or so that flowering plants have been around in any serious way, that both insects and plants co-evolved, as did hummingbirds and certain groups of bats, certain groups of other birds, honey creepers and those kind of things, to affect the pollination, to move the male gametes to the female receptive surfaces. So to do that, plants provide some rewards to those pollinators to enforce those behaviors. They can produce nectar mostly at the base of the flower, and these are simply primarily sugar solutions in which some protein, and in some other plants, actually lipids are part of the nectar that are produced. They're oil-producing flowers that certain types of bees are attracted to. So it's sugars, some protein, and rarely some oils. The pollen itself can be used as a food material. Usually pollen is produced in large enough numbers that bees and other pollinating insect vectors will actually feed on the pollen as part of their nutrition, but in so doing deliver other pollen to the female parts of the flower to affect the pollination. And as I mentioned, there are some oils that are present in certain flowers. Not a lot of succulent groups produce oil as a reward, a pollinator reward. So this is kind of like the bee's eye view of, in this case, looks like an Opuntia flower. And you have a receptive stigma surface, which is structurally strong. The insect can feel that as it lands on the flower. And then, of course, it has all of these stamens outside. And the stamens now, you can tell, are very, very, at the stage that they're very, very willing to donate and disperse their gametes, the pollen grains, onto the surface of the insect. Those of you that know about Opuntia flowers know that there are also these sensitive stamens there that if the insect touches them, the stamen actually moves and kind of whacks the side of the insect with pollen. And that is another mechanism that the flower has to ensure that the insect vector that has come to that flower is coated with enough pollen so as it moves to the next flower, it can transfer the pollen and hopefully affect some transfer of effective pollen that can grow down through that stigma and style. So you have flower presentations that, in this case, these are what we would consider solitary flowers. And in some of them, they are what we say sessile. They are attached right to the surface of the plant. So in this conophytum, you can see that the base of the flower is stuck right into the pair of leaves that there. The Huernia that's over here, the same thing. There's no real long stalk or anything. The flower is sessile or attached directly to the plant specifically. Another strategy is that if you produce lots of flowers and put them together in bunches to a potential pollinator that might look like a single reward source, a single source of nectar, but what that does is it clusters many, many flowers together in one apparatus, so that if a pollinator comes in and lands on there and it moves from flower to flower to flower, by having multiple flowers, it increases the probability of reproductive success. Each potential pollination is amplified by having more flowers in the same location. So Hoyas and the Asclepiad group of the Apocynaceae, Crassulas do this, having large numbers of small flowers. Haworthias, aloes, all of this strategy is well entrenched into many succulent groups, and that by producing larger numbers of smaller flowers, it enhances the potential for successful reproduction. That's what the benefit of having an inflorescence is, and the presence of multiple flowers. So we've mentioned that pollen is produced by the anthers attached to those stamens, and this is the uncertainty area where if those gametes are not moved by a pollinator, you will not have reproduction. So this is one of the costs of having sexual reproduction, and that cost is far outweighed by the genetic benefits of having that form of reproduction, simply because we have many of our plants are perennials that if they're not fertilized and pollinated this year, there's another flowering cycle coming up next year. And in the grand scheme of evolutionary biology, it's not a one-year thing. You're in it for the long haul. So those movable male gametes are important, but they do rely on some external source to be moved from the male parts to those receptive stigmas. The pollen itself can take on all sorts of different forms. Some of it can be a little spiky like this with openings in it. Most of the pollen will have some form of an opening. This is one of the gaps where the pollen tube actually breaks out of when it lands on the stigma. So it will grow from one of those apertures. Colpi is what we call them. Pollen can take on different types of growth forms. Monocots generally have just one opening. Most dicots, 75% of flowering plants have 3 or more pores in their structure. So there's a lot of variation within the pollen itself. And this is a mixture of pollen, just to give you an idea of some of the diversity of pollen in terms of their morphology. And separating very specifically, I want to accentuate this, pollination is not fertilization. They are 2 separate processes. Pollination is merely the movement of pollen from the anthers to the stigmas, and it can have both biotic vectors, the animals of some sort, insects, birds, bats, and so on, that would be able to transfer the pollen, or in some cases, abiotic forces can transfer the pollen. Most commonly among them is wind pollination. This is why all of our grasses are pollinated by and large. In some rare cases, aquatic plants have water pollination or physical movement of the flower, and the triggering of special stress reliefs in the plants can actually throw pollen into the air. So that's another one of these abiotic mechanisms. Another consideration with pollination that we really have to take into account within our succulent collections is that selfing, which means self-pollination, taking pollen from the same flower and putting it on the stigma of the same flower, is not going to work in many cases. Selfing is evolutionarily a dead end. Some plants will do it just to allow another cycle of reproduction to occur, but in general, that does not enhance or maintain genetic diversity. And if that happens too many times, there's actually a negative effect of that over the long haul in terms of the alleles, the genes that are present being passed down through those self-generation. So inbreeding in general is not a good thing evolutionarily. So again, one of the major modes of sexual reproduction is to enhance that genetic diversity, and inbreeding runs diametrically opposed to that, just counter to that whole premise of having this outcrossing take place. So we don't necessarily want to have selfing where you take pollen from the anthers and put them directly on the stigma. You certainly want to move them to another flower if it's possible, and that will enhance that genetic diversity. The actual stigma morphology, where the pollen is deposited, is also relevant to the kind of pollinator that is being used. So lobed, flat disc-shaped, somewhat pointed, or large area fan-shaped type stigmas would indicate some form of insect or animal pollination, because there's got to be a large enough surface for the pollen to land on and stick to. If we talk about feathery stigmas like this is what you would have in a wind-pollinated grass, for example. So many of our plants within succulents do not have feathery stigmas. They may have papillae at the end of a sticky stigma, somewhat like this lobe stigma, but they're not really feathery in the sense that they're trying to capture pollen out of the wind. To an insect, you land on the pollen, pollen producing organs, or on some other part of the flower and follow them in. The reward typically is down at the bottom. The nectar is often at the base, so that forces the insect to run by those pollen-containing parts. Here are some scarabid-type beetles that are feeding probably on the tissues of the flower and digging down toward that base down there. And in so doing, they're very clumsy, and they can get pollen on them. And as they fly from flower to flower, they will be able to transfer some pollen. But beetles in general are very, very clumsy and not very efficient pollinators. Neither are ants, for that matter. You'll find ants on flowers, but they don't really do a lot of good pollination. The best pollinators are hymenopterans, which are bees and wasps. They are among the absolute best pollinators. And somewhat less effective are butterflies. There's not a lot of surface on many of the parts that land on the flower of butterflies, but in some cases, the butterflies do make reasonably good pollinators. There are some lepidopterans, like hawk moths. The flowers have adapted specifically to those pollinators because of their long proboscis. So you have this long tongue that, to get to the reward of this Echinopsis flower, they're way down at the bottom. The nectary is down here. So for that hawk moth to feed on the flower, it has to take its proboscis and put that tongue basically all the way down the throat of the flower to achieve successful receipt of that liquid that's down at the base here. And in so doing, the male parts, you can see all of the stamens out here, and the female parts, the stigmas, are all what we say exserted. They extend beyond the surface of the flower. So as this moth is putting its tongue down, it doesn't realize it, but the anthers are dusting it with pollen so that when it moves to the next flower, the pollen is going to get wiped off by those stigmatic surfaces right there and affect pollination. So this is one of the many forms of very specialized insect pollination that can evolve. Similar to that is the fly pollination that we see in many of the stapeliads, whether it's Hoodia, whether it's Stapelia per se. All of these flowers are flowers that produce a chemical mimic to rotting flesh. There's a number of sulfur compounds that are produced and released. Anyone who has grown these in their greenhouse or in their house on the windowsill or wherever that has flowered these know that that smell is of some sort of a rotting type plant, animal matter that attract the flies, and the flies then are able to go past certain parts specifically and pick up bags of pollen like this, the so-called pollinia, which are attached by translator arms to this gland, which is sticky. That gland sticks to parts of the fly, and as the fly flies to the next flower, it will wipe off that whole structure, that pollinium, and affect pollination that way. So this is one of the mechanisms that, again, becomes very specific, pollinator to the specific flower. So insect pollination by far, for the vast majority of plants, the most effective. But other animals do assist us with pollination. Bats, for example, another common feature. And these are spectacular photographs from Merlin Tuttle from the Bat Conservation International, that he has studied this quite extensively, both on cacti and on agaves. This is the genus Leptonicterus pollinating an agave flower at night. And of course, hummingbirds, ocotillo, and various other pollination-specific animals interact with our plants all the time. So these reduce the uncertainty by becoming very specific. But if that doesn't work for you, then you can transfer the pollen. You can be the pollination vector by taking the pollen from the production parts, the male parts in these male flowers here, and then transferring them to the stigmatic surfaces of the female flowers. And you can affect that. Usually they say a soft camel hair brush is the best thing to use next to an actual bee. So this can be done certainly in captivity in your collections, but you really do have to become aware of the parts and the timing and receptivity of those parts. So understanding this whole life cycle, affecting pollination, allowing the tube to grow, and then eventually forming the embryo that will form inside the seed, this whole process, starting with the flower all the way through here, is something that people don't generally think about or see inside the flower. But there's a lot of activity taking place in those flowers that you never really can see unless you literally take it apart and have a look at that. Once the gametes are achieved and they fuse together, you form that zygote in that ovule down here, and then eventually that is affected by that growth of the pollen tube, the fusion of those two sperm nuclei from those pollen tubes that will grow down through the tissue. This is what pollen tubes actually look like. These are growing on a microscope slide in a special solution, a borate-containing solution. And you can literally see the darker staining sperm nuclei right here. That will become the fusion product of the next generation. This is a colorized scanning electron micrograph just showing a pollen grain and that sort of root-like pollen tube that would grow out of it to deliver the pollen grains. Here is an example of pollen grains that have attached to the stigma of some flower, but are stained with a particular material that under ultraviolet light fluoresce differentially. So you can literally see the pollen grains and the pollen tubes growing down through the tissues of the female part of the plant. This is what happens, part of the stuff that you never get to see when you do that pollination. Once that pollen tube enters this cell, then it can form the zygote and that central cell endosperm. Going back to that structure, it will grow in through the micropyle and then form what will be now the embryo inside of that, what we call the megagametophyte, the female part of the plant, down here inside the ovary. That whole structure then will become the seed. And in so doing, that process of fertilization, forming the embryo, immediately triggers through hormonal signals the input of food into that ovule to start supplying enough material to store there so that when the seed is mature, there's enough stored reserves of carbohydrates, basically sugars and starch, and protein, enough for the embryo to survive on. So inside the fruit wall, as it develops, you have the seeds. They are attached to the so-called funiculus. You can analogize that as an umbilical cord, and the nutrients literally flow into the seed and are stored inside of that structure in addition to the embryo, so that once that seed is mature, the fruit wall will expand and act as I mentioned earlier, as a dispersal mechanism. So there's different dispersal agents. Animals can disperse the seeds, the thing like sticking the beggar tick on the fur of an animal or something. Wind can blow them around. Water can move them through the landscape. And in some cases, animals with fleshy fruits will ingest them and carry them through their digestive system for deposition in other places. Some types of dispersal are taken very, very long distance. For example, Ripsilis baxifera is the most widespread, geographically widespread cactus, spanning essentially 2 continents, both Africa and South America, originating in South America, but through bird dispersal, we hypothesize it has been taken to Africa and spread sort of throughout tropical and subtropical Africa, all the way to Madagascar. So this is another long distance dispersal affected by animals based on the fruit type. In this case, they're berries that are highly desirable. There's other types of mechanisms. For example, ferrocactus, as the fruits are removed from the cactus plant, the pith, the center part of the fruit, is literally pulled away, and that creates an opening through which the mature seeds inside can fall. So if animals like raccoons, coatimundis, something like that, are feeding on these fruits and pulling them off the barrel cacti and dragging the fruits to the environment, they're literally dispersing the seeds through that opening. My friend Nigel Taylor in England was describing this mechanism, and because of this opening, this pore that was created by the removal of that pith material, he called this the pith pore example of salt shaker dispersal. So take that for what it's worth. Other dispersal agents can be birds, which will peck out the seeds and ingest the seeds of this Harrisia fruit that you would have still intact. And of course, there's explosive seeds that can break apart as the capsule matures. Many of you have seen this, both here. Dorstenia ejects the seeds from the fruit, those kinds of things. So there's all sorts of different dispersal mechanisms, even the ones that use incoming rainfall to disperse their seeds. And this is typified by the mesems, who produce fruits that form small cups, exposing the inside of the fruit. Of course, these are all the mature seeds inside here. This is Delosperma. These are closed fruits of Frithia and Cadia and Lapidaria. But in general, once the seed surface, the capsule surface it becomes wet, this now becomes a cup-shaped structure that incoming rainfall can literally wash out the fruits. We call it hydrocastic dispersal. So as rainfall comes in, the seeds are thrown out, and then once they land on the ground, surface running water actually can help disperse those seeds as well. If you get to, say, a lithops or a conophytum habitat that's on a slope, if you're looking for more plants, you always determine what the slope is, and you look down the sides of the slope and look for stripes of plants that generally tend to occur in those stripes because of this water dispersal that takes place. So there's literally splashing from clouds, splashing the rainfall into those fruits and throwing the seeds up to about 2 meters away from the capsule with an incoming raindrop. There's wind dispersal as well in follicles that would occur. This is in Pachypodium, that the seeds will have small hairs attached to it and the wind will blow them away. And then once that takes place, the actual seed surface may come into existence as well. In some cases, the rough surfaces can be used to carry seeds away from the plant by ants that sometimes store the seeds below ground, essentially planting them in their caches where they'll be able to grab the seeds. And in fact, some seeds actually have a reward attached to them. The ants that do this will feed on that little strophiole that's on there and leave the hard seed coat behind. And that helps disperse and essentially plants the seeds in an amenable environment. So there's a lot of variation in the seeds in terms of that seed coat and how some of it may be adaptive to sort of animal dispersal. Every one of those seeds that's mature and fully functional will have an embryo waiting to be activated, as I said before, by those environmental cues: water, amenable temperatures, air, and potential nutrients. Once you put those 3 things or 4 things together, you have germination taking place, and then the establishment of seedlings in the environment. So the embryo that's in there is waiting to receive those cues. If you supply those cues in a mature and functional seed, you will have germination. And then finally, once that emerges, once the root emerges and eventually get that plant body, the leaves and stem emerging from that seed, this triggers the end of sexual reproduction and the start of the maturation process of the adult. So you've all grown seeds of various types. You know, seedlings can vary tremendously. And again, that has to do a lot with the environment in which they are attempting to continue their growth. If they've been around for a few million years, the likelihood is that they're fairly well adapted to that environment. And the really benign environments that we supply as we grow seeds horticulturally really should be amenable to all of the germination. In some cases, some seeds are just difficult to germinate, like Dinteranthus. You have to have that right combination of moisture and heat to be able to coax them out of this dormancy that all seeds will have as they're released by the fruit. So there's lots of different forms of seedlings. I'm not going to go into that, but this again ends that stage of sexual reproduction that we're focusing on this morning. I want to thank you for the opportunity to talk with you. I apologize for the technical glitch we had toward the middle, which shut us down for a little bit. But I will be happy to take questions. And if you do have additional questions beyond what we can cover today, feel free to contact me at Iowa State University. And my email is simply rwallace at iastate.edu. That's Googleable as well. If you use that as a transitive verb, you can Google me as well. So happy to take any questions, and I'll turn it back to Gunnar.
Okay, let's stop. There you go. You have one more slide. Okay. All right, there you go. That's what I'm talking about.